that with all this started with, we had a reason why we started to have these meetings, but behind everything was José. That is, well, Paul is part of the Onuchic family, now in every sense, right? And well, José, as you know, well, there's a lot of people here that have been part of his group, and he has his history starting in São Carlos at USP, and then went to Caltech. From there, returned to Brazil, and then went back to UCSD. Now he's at Rice, but he has been pretty much everywhere, doing a lot of work, not only on protein folding, but also in chromatin, and he has lots of stories to tell us. So José, it's an honor and a pleasure to have you with us. Thank you, and you have the stage.
Thank you, Vitor. I think being the last speaker here, let's thank Vitor and Paul for putting all this beautiful event together. So thank you guys, it would be impossible without you. As I always tend to be late, I like to put that what I'm talking about is a little bit of a story that happened in the last 11 years when Peter and I moved to Houston, and also Aries, Lieberman Aiden at Baylor was moving there, starting his career. And so a lot of the stuff I'm going to tell here, I've just put all these photos here to make sure that you know that I haven't done anything, but I will talk a little bit about it. At least I'm going to tell my perspective of the story. You already had a chance of learning about it. You heard from Olga, you heard from Antonio, you heard from Vittori, so you have parts of it. I'll try to put my integrate view of all this together as we move along. I think that if we go, you all guys talked about chromosomes. I'm not going to tell all the story, but I wanted to point out that at least most of this talk will be talking about a resolution where a bead is 50 kilobases, right? It's good to keep that in perspective, that if you think about that most genes are between 500 to 5 kilobases. So we're talking about some things about like a larger than a gene. You heard it from Olga, the experiments are getting better, people are going to lower resolution. There are different things you learn on these different resolutions, but there are a lot of things that you learn at this time that's basically where spatially are genes localized, they come together. So there's a lot that you have about that structure. I always like to say that when I took molecular biology, structure was irrelevant. Everything was just the chemistry of having some transcription factor, something regulating. And it's interesting to come to these days that you come on this structure of having a look on that. So this figure here, you heard about high C maps, is this sort of contact maps, and it's not really just remember high C's, we extrapolate to a contact map, but they are just literally, as Olga told you, is a ligation experiment. So you know from ligation things are coming on space, and there's a little bit of massage and thinking about on trying to converting ligations into contact maps, but we're going to leave on that approximation as we come along. The other thing that's interesting on this figure is, at least on the human chromosome, became a long story, on the human genome, chromosomes tend to separate. They form what people call territories. This is one of the classic Kramer 2001 figures. Everybody shows that. There are much better things now. But as you're going to see, they don't necessarily happen on every species, right? So there are lots of interesting things that come from humans. And the question is how these things take place. What's the physics of it? Are these equilibrium process? Are these active process? You know that there's lots of things that are active. You heard a lot about motors, SMC, sort of structure maintenance proteins that are work on that and as we come along. Okay, I won't do that too much. But you see, as a theoretician, I always get amazed by this IC experiment. Because if you have a look on this figure, I will tell for fun. You first have to put formaldehyde or something else that cross-link these DNAs. Okay? Then you cut these things with restriction enzymes. Then you fill an S with biotin. Then you ligate. Then you cut all these pieces to figure out, like Olga told you very clearly yesterday, how these things that were supposed to be separating chain but closed in space, now they come into these small pieces here that you're going to sequence to figure out what comes together. Every time I look at this experiment, you can notice there's nothing that can go wrong here. It's just amazing the technology that has been developed that this works, and I just want to point out to say that there's a lot of smart people involved in all these things that have been done. And then, but the question is, people love to look at these maps. That's actually a 2D plot, but actually what you care is the three-dimensional structure. That's what's doing it. And one thing we learned, Peter is here a long time, is that basically the fact that you have a polymer in the top of this 2D map create lots of constraints into the problems. So actually, even if you don't have a perfect map, you can actually tell a lot about this three-dimensional structure because of this constraint of the polymer that's telling you that things have to respect a certain three-dimensional geometry. And that's the area we're very interested in moving forward. Another very cool part is that basically the spatial information is actually well connected to the biochemistry or to the epigenetics of the system. So here's just an example of a particular piece between chromosome 19 and in a particular cell. Here's a lymphoblastoid. Doesn't make too much difference what it is. And you see these patterns. And these patterns, basically, they just tells you that areas that are darker red, they're more in contact. The areas that are light. And here in the top, there are a few experiments where people have done changing epigenetics. These are methylation, acetylation experiments that people have done. And you start to observe there's a strong correlation between some of these experiments. Like, for example, here, on this case here, you see, for example, these blue parts well relate to some of these patterns. Other parts are not. And it's a very interesting say that now there's a correlation between this epigenetics, this particular modification you have on the genome. And why epigenetics is important, okay? A lot of people here talk about protein folding, but remember, protein folding are different sequences of proteins. If you look at the genome, every different cell, they have the same DNA. So what you're changing is how you're modifying these histones or how you modify these nucleosomes, how you modify these things, epigenetics that differentiate cells. Right, and we are going to see later that different cells are going to have different markers, and that's where these things get distinction. So basically, when we talk about here about types that we're going to call A's and B's, they are close. Most of the genome can be to first order thought about two types that people call euchromatin or heterochromatin. We call A and B. Euchromatin tend to be more fluid and it's more gene active. Heterochromatin is more compact, less gene active, tends to be more attracted to each other. But as lots of people work, particularly the group that Olga works now with areas, they have been finding that these two different sort of types are not sufficient. You have to go subtypes or compartments, you have to break them in a resolution, at least 5 or 6 to represent the data into these days. So what I'm going to tell you is about modeling these things is that we want to start a situation where we actually want to be able to predict the structure without using the high-C data. We're going to try to do a model, how we have worked for a long time together, where we start from, doesn't mean the other things we need to use high-C, as Antonio tell you about, on systems that don't have, but we want to start a situation where we start from epigenetic data. From this, we're trying to figure out these types that I told you. First approximation is A and B's, but actually we break into subtypes, so you have A1, A2, B1, B2, B3, B4. Actually, B4 appears very rarely, subtypes. From these types, I have now a polymer model where I can predict the ensemble of three-dimensional structures. Chromosomes are actually much more fluid than proteins. They tend to be much more dynamical, so we really have to talk about ensembles. We cannot talk about single structures. Okay, and so that makes the problem. Now, the Hamiltonian we'll be talking about, let's talk about the first part. You heard a little bit about Antonio, but our model starts, we have a homopolymer term, a term that we call a spatial interaction term that tells A's attract A's, B's attract B, A's and B's repel each other. Actually, B attracts stronger. So there's sort of a polymer term. And there's another term that we call the ideal chromosomes that tell you that beads that are closer to each other in sequence, they all tend to be more attractive. I'll talk a little bit about it. They're loops. But a lot, if you stay on the 50 kilobase resolution, they're not that important for the overall structure. They can be included or not included in the model. We can do both ways, depending on where you are trying to go. So let's talk about this first part here. And then I talk about the second part here. That's how we go from epigenetics to these types. But let's assume I have a sequence. So my sequence here, I'm going to break my entire genome into this sequence of beads. Each bead is 50 kilobase. Right? And they're going to be associated to one of particular these types. But remember, these are modifications to the genes, the same DNA. Okay, as we move from here, here's the same Hamiltonian I told you before. We can now try, what we can do is, we're going to do a mix of information and physics. We believe we have the right physics, but I'm going to use some structural information to learn these parameters, to learn what should be these interactions between types, and to learn what should be the interaction about this ideal chromosome as we come along. So as we move, and I want to tell you about the ideal chromosome, this is a term. If you think about interactions into the sequence, these interactions in sequence tend to form helical structures, right? Think about proteins. You are used to it. II plus 4 give you alpha helix. Right? So this interaction, this sequence, create this nature that we call helices, and helices of helices is sort of liquid crystalline nature of the chromosome. And actually is one of the reasons, because you have this high density of local contacts that minimize the number of knots. As chromosomes, you know, they have much less knots than they have a run of heteropolymer. Okay. So it's important to do. It's an idea that Peter came a long time ago, is now has been included into this model. So now what we do is basically, we look at the cell, this lymphoblastoid cell. They have all these different chromosomes. We get one of them. We chose chromosome 10 because they have a lot of the patterns that we want to learn from it. And we learn the parameters. Okay? These are standard sort of maximum entropy ideas where you assume everything you don't know, you're maximizing disorder, and you try to learn the parameters. Now, maximum entropy is a good idea, but it's important to keep in mind, people always say, if you want to maximize, the best ensemble you get, given the information you have, is the one that comes from maximum entropy. But you have to remember one thing. If you don't have enough information, the best may not be sufficient. As I like to describe, when you have a research group, your best student may not be good enough. So you have to remember that you have to have enough data, enough information in order to get things done. But anyway, these are just learned. We use a single chromosome of all the chromosomes. After that, we fix the parameters. The parameters are not changed. And you're going to see that this set of fixed parameters into this energy model, this Hamiltonian, is going to be good for all the other chromosomes. It's going to be good for different cell types. So it's a great thing to move forward. Now, there's a second part. The second part comes from the fact that basically now, how do I learn from my epigenetic information, most of it chip-seq, but there are lots of other data coming these days. There's attack-seq, there are lots of other data that you can actually use into these things in terms of learning these types. And the stuff we're going to use is we're going to use the same idea that basically we did on covariance. That's actually the heart of AlphaFold. They got a Nobel Prize today. That basically you have a bunch of sequences aligned, and you look the following. You don't look like how residues change together, but you see how they change in pairs. And by doing that, you can figure out if they are making a native contact or not making a native contact. It's one of these great tools. We do a fake sequence, where our sequence, that being a protein sequence here now, is we get every locus on my entire genome, and for a locus I put the type I want as the first residue of the chain, and all the experiments as the other parts of the chains. So now and like I said, before I tell you a little bit about it, I will go back there. I want to say why we believe this is a good idea. This sort of, this GM1278 that I told you is the lymphoblastoid I'll talk about. And here's, for example, the pulmonary cell, particular cancer pulmonary cell. And you can see the maps are different. And you see that these differences are actually correlate to the differences. I don't know where this pointer is. It disappeared on me. It's very weak. You can see that basically in the top there are major difference, and the patterns actually are correlated by these differences in epigenetics. So if I move forward one slide, it's exactly what I want to get here. You look for each of these experiments. The points here tells you how strong is the signal for each of these markers on each point. And now, what's nice about doing covariance? I'm not looking, for example, how this particular type that I have here is related to each experiment, but I'm also seeing how experiments change together. So by using covariance, I'm also really improving the capability of the system. And now I can start to go what I call, and for the people who are from physics here, this model of covariance and all these things are using in proteins it's a beautiful model because since it's pairwise, and you can say how much a residue falls into what people call a Potts model. That's basic Ising's model, who is a spin of many, many different orientations. In case of proteins, we have 20 amino acids for gap delicious. But there's entire statistical physics that you can use for this model that's already in place that you can use here. So the bottom line is, when I put all this information together, now what you see is, given a particular chromosome here, I'm showing a particular case here, you can see that basically these are the types, A1, A2, B2, that you get from high C. This is what you get from Megabase. You see you do quite well on the predictions. As expected, if you look at this prediction, you can see, if you look with very sharp eyes, where you run into trouble. When we decide that we have A's and B's, or A1, B2, I'm getting a problem that's continuous in terms of all these markers, and I'm discretizing. As soon as I get discretizing, you can observe that on areas where you have larger fluctuations too fast, that's when I'm going to make more mistakes. That's not a surprise, right? That comes from the approach, but that has very little effect on the three-dimensional structure. So by using the first part of the model, that's basically coming from ChIP-seq, we can get the types. And this is just something you heard from Antonio yesterday. Said, if you don't want to deal with a problem where you approximate yourself to a few types, and you want now to do an energy function where you completely invert the entire matrix and get with thousands and thousands of parameters, I still can come to the first eigenvalue, and you see the first eigenvalue gives you the A's and B's, and you see that it does quite well. So this description is not a bad one, right? You see what you get A's and B's if you use here, or if you just the full inversion, tells you that actually approximation into a few types is not bad for chromosomes, okay? I won't tell the details about this method. You heard from Antonio how you use this on bacteria, on other systems. We've been using on yeast with Vittori, but based here I'm going to stay most of this on the humankind, or mice, where you can live with types. So the nice thing now is we have a method that's predictive, has been out there. We have been using for many things. We are used for many cells. There's what we call the ENCODE database, and we're having a lot of fun predicting all the structure of these chromosomes. It would be nice as they start to come out and see how well we did. Basically, we're really exposing ourselves, but that's the fun of doing science. We are just dumping all these structures into something where there's no structural information in there. And I think it's sort of an interesting game to be played, but you see you get the structures. Now, the nice thing now is that basically, as I get this ensemble of structures, I can go now for the particular cells that I have high C data. I can compare if my predictions are in agreement with high C. And this is a comparison. As you observe, just for, this is sort of, the top part is our prediction, the bottom part are experiments. As you can see, around the diagonal, you have lots of dense contacts and you do quite well, but just comparing around the diagonal is not good enough. If you really want to compare three-dimensional structures, you want to compare on areas that are far from the diagonal, and you can see they'll do quite well. What do I mean by doing quite well? You can judge as well as I do, but as you can see, I can guarantee you 3D structures, when you have this level of similarity, are very, very similar. The ensemble is very, very similar, because you have polymer restricting. You see the areas of contacts. You see there's a level of more noise, less noise. You guys love to hear about AI and prediction. What's data, what's noise? We're actually doing a lot of work. We have lots of collaborators. Peter has been very interested on that. Vittorio keeps shouting on my ears that say I'm fitting noise. So you see, there's a lot of interesting stuff going around. Vinicius and Antonio keep telling I'm wrong all the time, but I win most of the days. But you see, it's an interesting game as you start to deal with data and you start to move forward as we look at this game. But it's nice to see that basically the areas, if you just put your eyes on areas that have large number of contacts, a small number of contacts, really associated with this three-dimensional structure, predictions are quite good. So we're very happy with that. Okay? And this nature of the chromosome being quite good and being very robust is what allows us to also be very confident for that lovely talk that we heard about the mammals yesterday by Olga, that basically we can come up from data that's not that high quality, but we know the right physics that's coupling these things, and we can come up with structures on these sort of extinct species structures of genome. So that's nice. So as I am here, we have, you heard yesterday, we have, Antonio was more in details, we have all the software in there at different levels of sophistication, both for the simulations and both for the predicting the types. I want to go back one slide and just remind that basically, since I'm not going to show a slide at the end, in addition, the National Science Foundation has been supporting us forever on this effort, money from the Welsh Foundation. I want to point out that AMD has been very generous on providing computer power for us. It's great. Bo is here. We can joke with Raphael that says basically the nightmare, and Vinicius can tell us, is to get out of CUDA because not an NVIDIA one. There's a lot of work that goes in that. That's part of the game, just for the curiosity of the people that are interested in that. So this is just to show you that basically now we can start to apply that for different cells, and actually these models are actually reasonably good, as long as you can figure out just the different types. So, what becomes the game now? The game now, you can come and say you're cheating. And every time you deal with data, there is a degree of cheating. Basically, you're trying to copy the information you got from there. Clearly here, we are not really doing dirt learning. You're just using the data in order to learn these parameters, a very small set of parameters. But it's nice to go and to compare to experiments that you have no link to it. And clearly on the system is the imaging data. Imaging is becoming a very clever way of looking at this information on this genome, and now on imaging you can say we never used any of this imaging data to adjust our data, so it becomes a great thing to go about it. Shuai Zhang did these experiments, and the first part, I will tell you about the experiment. The first part, she got, on their first experiment, she didn't do the full chromosomes, just these particular pieces of the chromosome. I'm going to tell you about it. And she looked at the structures in the experiment many, many times. And she was very annoyed in the beginning because she was not getting a structure. Every time you do the experiment, you get a different structure because actually, like I told you, these things are much more fluid. You have an ensemble. So you don't have a single structure to compare. I won't tell you about the experiments, but they are fairly complicated experiments because you see that you have these markers, you hybridization one part, you measure one part, then you measure the other part, On each one of them there's a wash, so you can always wonder. So there's a lot of effort on doing these things. These are not dynamic experiments. That's based when you get a particular sample, you just freeze it, and you get that particular structure. Just remember, these experiments are becoming very common these days. Aries group and August group actually are doing some of these experiments. And we are working a lot on doing the interpretation of these things as they come along. Now, what's nice is that basically on this first experiment, you're looking at two particular segments. And if you look at the segments, these are the segments that are about a couple megabases, okay? If you think about our units here, 50 kilobase, you're talking about 40 bit segments in the kind of resolution we are doing here. So you can keep that in mind, just remember. And what you observe, there are two segments. This first one I call segment one. And the segment one, the top, let me just see if this thing gets, okay. The segment one, as you're having a look here, was one where you see a little bit of more structure. Segment two is more disordered. It's not a surprise that this one falls much more, the top one falls much more on the port active area of the chromatin, and the other one is the inactive area, but that's the data, they got it. So now, at that time in the group, people start to have a look on part. This is the top segment, and what Ryan Sheng did, he observed that if you get segment one actually fall on a case that was a closed dumbbell, was more collapsed structures, and the other possibility to have open structure. You can see here the numbers are heavily biased toward the closed one. That means there's a little bit more stability, but the system is still opening and closing. If you look at the simulation stuff, it's more or less the same thing. Not exactly. If you have a look here when you do the free energy profile, you can see the experiments a little bit more likely to be open than the simulations, but that's details of the data that we're using here. Okay? And I haven't told you guys for too much time. There are a few things that we have to include on the model, but that's a good time to remind you. Remember that the ideal chromosome, actually, most of the term, you heard a lot of talks about loop extrusion and mechanism of motors. We are a little bit agnostic about it. We're just assuming that this compactation, this term, what we call the ideal chromosome, we just fit to the data. Motorization is a probable cause of it, but we may have other things that may work on doing that compactation. We are just putting the model. So when we come to 50 kilobases, we have a potential that make that compactation. But remember, that thing is actually coming from an active effect, right? So we're effectively putting in the system. The temperature we run the simulations here, what we call temperature 1, for lack of a better name, just to be clear, is the temperature where you observe the fluctuation of the chromosome that agree with experiments, right? Because they're active process here. So we have an effective temperature into this system, just for the aficionados doing this game, and exactly the temperature is not a surprise that we may not, depend on experimental conditions, agree perfectly well, but we agree quite well. Okay, there's a difference about 4 KTs. That's basically what appears to be activation between the closed one and the open one. 4 KT is also a magic number for us in a complete different situation. On the homopolar model that we have actually for this potential is actually a soffit model. That means the chains can cut to put the effect of top isomerization. In the model, if you run the simulation with our software, you can put as much as you want at that number there. Basically, we have this number about 4 KT. What's very nice is, although chains can cut, you're going to observe the equilibrium state of this model tend to be the very unknotted state for the chromosome. Okay, what's nice about it, things reasonably agree with each other. You see here rates of gyration. You told you a little bit about the difference of the potential. So the numbers of these ensembles agree quite well. And what's very cool is that if you look the final results on this dumbbell, is that actually these things are open and closed, but if you have this link between the two ends, This link is where a lot of genes are located. So in a sense, this is the first time in this very primitive one, or very rudimental one, where you can associate this structural information towards function, right? You can see that this opening and closing is going to expose or not expose these genes for function, and it's kind of a nice result to see. I don't want to say that this is a prediction. I want to say that this level is much more an observation, right? But it's very nice to observe that in the other case where you have no genes on segment B, they tend to be just a disorder collapse state. You don't see these sort of open and closed states that we see from the other case. So this is kind of nice, puts you a little bit put the system on reference on how the systems may work. So we're very happy about it. So the question now, you heard a lot of different talks here from the mammals, from other things. How can we move forward into the system? And I don't want to, since I'm the last speaker, I don't want to go forever. I'll tell you a few stories, few short stories, just for the entertainment of seeing how powerful these methods are compared with all the great experiments that we have out here. Here is just another case we are doing here, basically. These are three different cell lines, and we look for a particular region on the cell lines where there is a major difference. And you see here, observe that UVEC becomes B-like, heterochromatin-like, while the other ones. And this particular area tells you that when this area are gene active, tend to be much more going to the surface of the chromosome. Then when you're active, you go inside. That's kind of nice to observe that although the cells are not that different, they have this ensemble, you have this sort of behavior appears very naturally from you, that basically gene expressing parts tend to get to go much more outside than the ones that go inside. I want to just show 2 slides that basically, and I won't go into details. Now in the game, not looking at a single chromosome, but looking at many chromosomes, right? And we are doing the simulation of trying to do the full nucleus. There are several systems we're going after that. The yeast is one of the ideal systems for Augusto to actually look the full system. But even the human one, this is an approximation because these things we don't have laminin, we don't have the effects of the surface are weak. But one thing I want to point out, for some reason my computer there doesn't like the move here. This is a simulation. They're running about on the time of a cell cycle. And you see, this doesn't want to work, but that's okay. Oh, let me go back. But you see that these balls here are the center of mass of each chromosome, and you see on the time of a cell cycle, they don't do it. They cannot completely move around. What tells you that based on a cell cycle, chromosomes can actually average over the entire system. You can say you have an equilibrium ensemble of them, but that's not true for the full nucleus. The entire all chromosomes cannot move around. So that's the only information I want to point out for you guys here to remember. Now, a few years ago, 2 or 3 years, Olga? Yes, 3 years ago, we started to look not at a single species. One of the great things that Olga has been responsible for has been what we call this DNA zoo, that based not only she's being able to sort of sequence every single mammal species and other species, a lot of eukaryotic species, but also we have been able to look at the chromosome structure for all these different species. Right, so it's very nice to start to compare. And one of the clever things is that basically in humans, like I told you before, or high eukaryotes, chicken, mammals, they tend to have this structure where you have chromosome territories. Right? This chromosome separate and do it. That's not true on many other species that are going to go around as you go down to mosquitoes or to mushrooms or whatever, where you start to have these things where you start to have this organization of centromeres or telomeres instead of having this organization for the nucleus. You have these things where you call situation you have centromere clustering or telomere clustering, or a polarization where you have the telomere and the clustering polarized in the 2 ends. So the question is, how do you go from one to the other? Why some species are here? And on this paper we published 3 years ago, in collaboration with Aries Group and Ben Holland in Holland, the clusters in Holland, we start to observe that some species, I can look at here, but I'm going to show another slide because it's easier to see. You start to see that basically different species fall into different classes. If you have a look, if you have the humans one is here, you see that clearly the chromosomes are separate. You see each chromosome have lots of contacts, but interaction between chromosomes is very weak. That's what we call territory formation, right? They tend to be clustered. But you go to other species here, where now we start to see centromeres coming together, for example here, right? You see now the points are. And the bottom line is just like I told you before, if you look at these maps very carefully, you can almost break them into 4 categories, the ones we call the ones that actually are forming territories, and the ones that don't form territories that either polarize these things, have centromere cluster, telomere clustering, or the axis. Here is a different figure where you average overall the structure to give a better situation. As you can see, when you have chromosome territory, you have one chromosome, the other chromosome, very weak coupling between them. When you have cluster centromeres, you see the centers coming together. When you have telomeres, you see the ends coming together. And then you have this axis when both come. So it's a different maps, different species coming together. What's very interesting that you observe, and observe from our simulations, is that going between these regimes have a lot to do with that ideal chromosome. And the idea of chromosome, I told you, that term that tells you if your terms of the sequence get stronger, they depend on the degree of motorization. It's how these motors are able to compact it together and bring together is what form these things to come together. And it's very nice that basically when you have strong motorization, you really have territories, and as you go down in motorization, you start to see these other mechanisms that go out. So there is an interesting question, why is that the case when you look at your systems of having one with more motorization, one with less motorization, on different species. So that's what we did. But we can actually classify all the species on this category. So Sumitao Ramachari, together with Antonio, Vinicius, everybody here, put this simple model together. It's actually even a simplification of what we call micron or potential before. It's just add now a competition between this compactation versus centromere attraction with a little bit of lamina. But the most important thing is these two competitions. How much motorization you have versus centromere attraction. And when you work this model around, I can go into details, but it's getting too late, you see the following situation. With weak less-wise compactation, you get a lot of centromere clustered, and you have weak territories. Right? As you increase motorization in the model, you see now the centromeres get separate, and you get territories being formed. And this is kind of nice, because if I come to this slide, these are the predictions. What Ben Rowland did say, if that's true, I can get a human chromosome. If I weaken motorization, I should change the regime. And that's exactly what you observed, as you can see from this plot. The wild type, you have all these centromeres going around, okay? And when he depletes, you see all the centromeres coming together. So in a sense, what we like to say is by depleting these motors, we have humans becoming mosquitoes. Right? So you have this transformation of species by weakening motorization, right? And this is going completely one-to-one agreement from strong territories. To, see these points here that tell about centromere attractions here, and the experiments give you the same story here. Okay, so this is a very nice story. I think this is a story that really starts with the DNA Zoo, big effort by Olga, this other collaboration. I think this paper really brought together the fact. Now there is a very interesting question in all our hands that's to ask why these things are done, how these things are important for different species, what's the functional aspects of that, and that's really where we have to move forward as we move along. So now, since because of my guilt yesterday of not giving my talk at the appropriate time, poor Vinicius was cut out. taking out of the schedule in a complete unbecoming behavior. Let me tell you about a great result that comes from work that he's doing now. That's basically all this data. Most of the discussion we did now, we are talking about chromosomes at the interface. Right? That's where you leave 95% of your time. As you can see, if you look at this marker, this big brown circle going around. But there's a lot of stuff interesting that happens in the middle, right? And people start to do experiments, say, do these terms that I have, these interactions between A's and B's, these ideal chromosomes, how they work as we move along. So on this particular experimental paper, people start to look at high-C maps at all these different stages during the cell cycle for chicken, and we sort of start to look at this data as we move along. So basically, these are beautiful experiments. Olga can give you a better answer than I can, how good the synchronization on each one of these states, right? There's a lot of stuff to know. Maybe you have a mix of data. So I'm not telling you everything is golden here, but it's a very interesting story to make sure that you're measuring these things. But even at this level, there are lots of interesting stuff that you can get out of it. So if you look at different stages during the cell cycle, you observe that basically they were able to get the high C as you move along. And even at this level, although this is not the most high quality high C maps you can get it, you start to observe that as you're moving down here, this diagonal starts to get thicker. You have more contacts around the diagonal. That appears there's an increase in motorization, even just by look at that. I'm going to show better data, but just look at here, you can already have a feeling. You start to observe that, see if I can get a little bit of red here. No, I don't. You can see from the point, you have long-range contacts. You go to the other end, to the mitotic end, you start to have less long-range contacts. So you're going to have less territory formation. Right, there's a natural way, less compartments. These things don't get that compartment as you can get it. And actually, you can see that going down here, you may have multi-diagonals. It's not you have a diagonal, then you have another parallel diagonals going around. That means you have some more local structures being built as you're moving along. So this sort of come out of the data. Here is one thing that tells you basically that's the interface. You have your map, your high C map here. That is second one that tells you basically you have lots of contacts, both around the diagonal and also far from the diagonal. So you have a very circular kind of chromosome. Okay? You observe that basically the contacts here tend to be a nice power law, not perfect, but it's can be fit by a very nice power law as you have seen a lot of interface. And then when you try to learn the potentials from there, you see that you can have strong interactions between A's, strong interactions between B with different signs. There's actually a very strong phase separation between them. The potential we learn give a very strong separation. You have red strong and blue, then you get of these interactions. S, when I get down here, observed this potential now here, you see that basically you have much less long-range contacts. Observe that the contact formation, you have a shoulder in the middle, very non-conventional for the interface. You see that your AB nature start to go down. You have a much more stronger effect of the motorization. And actually you have motorization at two different scales that come from the map. And the A's and B's are very weakly separate. There's not that strong separation between parameters. This is something you're learning from the data. And actually, you can do the entire system. You can see how you move from one to the other. Here's our typical structures. And you see, use the same kind of potential, just using these different parameters that now we're learning from these different high C maps. You see now you go from something very spherical, from something that's more cylindrical-like. You see some things more disorder, something that's very helical-like. Actually, the helical-like is kind of very interesting, because I told you if the ideal chromosome gets stronger, you expect it to be the case. So Vinicius worked very hard to make this point, and actually made a clear case. What he does is, for each one of this particular structure, he did some annealing to see what are the low energy structures in each one of these bases. Remember, you have this very dynamical base, the chromosome moving, but I tried just to anneal down in temperature, just to see what's the main attractor of each one of those. And when he does that, you start to see that as you go down, you become much more helical. Actually, it's even more interesting than that. Let me go one more, and I go back to the structures if I come down here. He has this parameter, we call these order parameters, that tells you how ordered you are as we move along. Interesting is, although you can learn a lot about this order parameter, you start to see you get much more order as you come down. You see these natures of, it's much easier to look at the Fourier transform of these things. And for the Fourier transforms, you see that basically these low energy structures around here, there's a helices. They have this helical nature, as I told you before. Okay? And you see the typical helices is about 0.2 megabase of the pitch of the helices, right? But as you come down to the mitotic one, not only you become much more cylindrical, but now you start to see you have two helices, right? You see that now you have a helices again to around 0.2 megabase, but you have another much longer helice on the order of tens of megabase. Right? And actually, if you want to see that, that's basically filtering the low frequency one, just get the low frequency, filter the high frequency. You see that basically at the level of the interface, if you take out the high frequency local helices, you have kind of a disorder system. When you go down to the mitotic, you get these things to become much more helical-like. So this idea is what I told you about helices of helices, they really become totally apparent, this liquid crystalline nature as you go to mitotic. It's very interesting to know how all these transitions happen. Clearly, at this level, also you notice that your ideal chromosome is stronger, your phase separation is weaker. That means you're really increasing motorization. As expected, you know that. As you go down to mitosis, you start to increase the levels of condensin 2 and then condensin 1. You increase this level. It's very nice from the data. You can see this nature of the cell cycle as we come along. This is actually something that Peter cares about and I care about and everybody cares about. The question is, how about chirality of the systems? Is chirality important? There's the problem. Chirality is always a good case. Can you create chiral traps or not? The high C data tells you nothing about chirality. Our model is chiral free, so we don't have anything. So one thing we notice is that basically you have a lot of chiral traps here. If you have perfect polymers, it will take thousands of cell cycles to get these things to go out. But the system is much more complicated than that. You have other motors going there. You have polymerases. You have supercoiling. You have topoisomerization coming in. So it's a very interesting question all these times and these dynamics of the things as you move along. I just want to put you for the flavor about the things you have to think about it as you're moving towards functional aspects of that system. Now to finish, because I'm out of time, I had 4 or 5 more topics, but I'm not going to do it. Is the mosquito. Basically, here in Brazil, we know we have a very close relationships with this mosquito, thanks to dengue. So it's sort of something that we have to keep in mind. And this mosquito is kind of nice. Vinicius came it some time ago and had a look on it. And you look at the high C map. And this high C map, you just look at the high C map, just look at it, you understand these things that basically you have your diagonal, but you also have an anti-diagonal interaction, if you have a look. You have also telomere attractions and centromere attractions. So just look at the high C, you can start to guess what is your genome. You look at the what I told you about the different situation we have. What's kind of nice here, as I'm going to show you, is that you start to see now these situations that you don't see on the human chromosome. So you believe here in a sort of zero-order model. I draw this cartoon down here. I'm telling you, centromeres are coming together, telomeres are coming together, and you have this antediagonal, they have probably the arms of these chromosomes are coming together. So that's a cartoon. But actually, and this is very different than you get from the human chromosome. I just put the human here to have an, there's a couple chromosomes per human to notice that here you have very well-defined territories, very weak coupling. See, so it's kind of cool just to compare and to have a look the difference of these maps compared to humans. So Vinicius tried to model it, and it's kind of nice. I'm going to go fast to go around it. Let me just go over stuff that basically, here's a quench simulation. You see that this chromosome is much more helical, has this sort of long-range helices, even at the interface. Right? It's a very interesting question. Why? You see, if we look the human, that was mice, or right, or human, the same story. In order to get this level of helicity, you have to go down to very mitotic-like. Right here, you start to observe, just like before, you see that you have this sort of 0.6 megabase. The human, we get about 0.2, 0.3. This is about 10 megabase for the other helicity. It's kind of a nice system. Here's the order parameter plot. So it's sort of a very nice story to see how these things look. And I just wanted to say one more point about this for people to think about it. What's very nice, we have a lot of effort. We have here our force expert, Rafael here, and a lot of people are talking about force being applied on systems. And that's a system that's very easy to do. Here's the poor person's approach to force simulations. What's very nice is that if you get a human chromosome, and you start to pull it, you start to unfold from the ends. But nothing too interesting takes place. You see here you have low force, increase the force, you come up. However, if you do that to the mosquito and you apply force, you see to really extend these things. This is much more mechanical sensitive. Okay, I think these are things that are very interesting about this interaction between the entire function and mechanical cues, how they come together, how these things are putting together. Okay? So with that, I think I'm going to skip this entire part. I'm not going to tell about the bacteria. Also, I'm going to tell just one thing for the next talk we invite. There's a very interesting thing about bacteria that basically you have replication and segregation happen at the same time. Right? Different than humans where you have a very precise cell duplication. Here you actually, if you are in the exponential growth of bacteria, these things are duplicating and segregating at the same time. But I won't have time to talk about it, but there's a very thing how we can use high-C data and all this information to go forward. And I just want to finish here and tell you about the people that have been involved in protein folding and the chromosome work with me for all these years. Without them, nothing would be possible. I love to say when people one time ask me, say, if you travel so much, who does the work when you're traveling? I say the same people that do the work when I'm not traveling. So everything would be impossible without them. And I thank for your attention and patience until the end of the day. Thank you.
So since it's the last session, so we have time for questions, other questions we want. Questions? So I can start with the first one. Of course, Jose, you already know where I'm going. Chromosomes are way tougher in terms of, because you have this liquid-like character. My question is, we clearly see that how you go, you have these domains and you have this very nice simulations in which you see the domains going on. So can you cook up kind of what would be the analogy that we would do in terms of having an order parameter that you can put the entire process as you go from one to another? And what the story would be, well, if you have that, or if you could have this reaction coordinate, or even if it's a multidimensional parameter, you could, or characterization of these things, eventually you could try, well, you know what I plan to do.
Anyway,
so how would you connect with all the energy landscape that we've been talking about?
Couple things. When things are less structured, in some ways they are more complicated, some ways they are much easier. Right? You have much less this sort of trapping situation and all this, so there's things you can do. The interesting question you always have, every time when you ask about order parameters and things like that, you look at systems on very high dimensionality, and you say, can I collapse down to a few coordinates and tell something about it? Okay, and that has been a very interesting question of what is the degree of, how this degree of order comes into chromosomes. And I think we can get different people with different opinions here. But if you look, most of the chromosomes you have a look, at least the human ones, you tend to have big packs of the blue coming together, and then you have smaller bubbles of the A's going around, and these things are much more dynamical as the A's come in and come out. So I think in terms to describe the system, in terms of these major cores and how they move in and they join together and come together is the way it should be done. We have been trying a few of these things. It works. Sometimes if you just use the sort of the conventional cues that we have on proteins, can be my cue, Peter's cue, they tend to be too precise to look at chromosomes. So you may have to have things a little bit more diffuse, but we are trying that. But there is a very interesting question. Basically, I show you some cartoonish views. We observe that, for example, in most chromosomes, at least the human ones, the active part tend to be outside and inside, so you can have models that tells you where are the A's, where are the B's. There are lots of organization we have, but we don't have yet, and maybe we may never have, a few defined comfortable order parameters as we have for proteins, since they have a more well-defined structure. But it's a very interesting question to tell you to do that. The other thing that we have friendly disagreements is how to measure similarities between ensembles, right? It's basically how deep you have to go, what make to say two ensembles are similar to each other, and that's a very interesting question, particularly when you relate these things to function.
My question, I don't know if you know how to answer that, but I was curious about the energetics when you're comparing, like you're talking about a lot of the mosquitoes versus the human, and I'm like there's so much more motorization, so much more compactation in the human, so how much more energy our cells needs versus the mosquitoes to do that process? Do you know the information? Is that somehow, makes sense, my question first?
It makes sense. The question is, why at different levels we have different levels of motorization, right? And basically, and that in some way it's kind of obvious. As you start to get more mitotic-like, and you try to compact these things more and get these things dependent, you have to increase the level of motorization. Mm-hmm. Right now, why the mosquito shows that at an earlier level, it is a very interesting question. Why do you rather have centromeres coming together than have territories? These are very interesting questions. So I don't think I can answer. We can really say that they appear to be on a level of motorization already interface much higher than humans are. Right? But that's more an observation than a conclusion.
Hello, José. Very nice talk. I like in particular how I think that your work is driving to a connection between different scales, in the sense that you find a relation between motorization and territoriality, compartmentalization, that we know that they are very different phenomena, but somehow they are connected. I love it. But I have a small question regarding to the mosquito pooling. Like you show the human that indeed is very fluid, and the mosquito that is more solid, and you connect it to motorization. How does it relate to this, experiment of John Marco, where, for instance, they take out chromosome in mitosis, and they look very stiff. Like, can your simulation from IC reproduce this kind of stiffness?
Okay, we haven't measured stiffness unless Vinicius has measured, and I have ignorance about it. But clearly they're more stiff, indirectly. Otherwise, you wouldn't have this mechanical response that you observe. Okay? So the fact that with the human, when you just do this very simple experiment at the end, when you pull them apart and just the end comes, tells you there's much less connection between these parts. Right? The mosquito, clear, when you pull at the end, everything else comes together. That's already to a certain degree. These things are coupled and tend to be stiffer. We haven't done more detailed experiments than that, but I think there's an evidence. Do you want to comment on that, Vinicius, a little bit?
Yeah, so we actually have work with one of our undergrads, Ben, that he actually did some pooling and compared with the micro experiments. And indeed, the interface, they are more fluid than you can actually get. You can really rapidly go to a non-linear regime, then things get more complicated. But with the mitotic, it's more or less compared to the mosquito, where it's more stiff, and then you can actually see the formations. Yeah. In silico, ours was in silico. Yeah. So yes, so we do not directly compare with the, because it was a different length.
But should be
yeah,
done.
Could be done, yeah.
Thank you.
Yeah, beautiful talk, Jose. I have two questions. One is, you guys have been learning so much over the last, I guess, decade with this. So with both structural knowledge, the three-dimensional and dynamics and so on, does it sort of, do you see an angle of maybe engineering or biological sort of to connect it to function that you could start to control like from your studies or your simulations, you could start to understand how you could control genomic function and so on. And my second question is about the helices. I think I remember from Bin and Peter's paper that there is some argument for why they are so ubiquitous, but I can't remember now. Like, I mean, I understand the data inversion, but is there some statistical, mechanical argument why there should be so many helices there?
Okay, so there are lots of interesting questions. Let's start with number 2, then I go to number 1. Okay, the fact that you have this sort of ideal chromosome term and this helical tendons, I think one of the things that appear to be connected to that, I'm not saying it's the only thing, is the fact that really reduced the amount of knotting inside the chromosome. Right? So if that's the only reason, I don't know, but you really want to have a very unknotted chromosome, because if you do some function, you want to be able to pull one part without having to pull the entire thing apart. So that's an interesting question. If there are other reasons for that, it's true. The second part of that is that basically as you increase the compactation, helices are going to appear naturally with it. It's a natural way that it's going to appear. Peter can give his opinion about that. So it's not a secret that basically, you see when you have to become mitotic and you have to be highly compact, this helix is a natural way of really compacting the systems more than you have in any other sort of random rearrangement. But I will let him comment on that later. Let me finish the second question, you can comment on that. Second, in function is very interesting. If you look on the case where we show about José Zhang data, you see that basically you start to see that you have this opens and closing of exposing. So appears there's a double interplay into the genome. At one point you have to have these things spatially protected or spatially exposed and all that. But at the same time cannot be too rigid, because the things have to be able to duplicate, have to be able to transcribe, have to be able to do all that. So the question is, where's the sweet spot here? I don't know. But there is a question that now we know in many cases that basically this sort of structures are very interesting. Olga can even tell you more details, for example, about the genes on the elephant, like the hair genes, where they go in the elephant versus the mammoths, because one is active, the other is not active. So there are lots of interesting that comes from that. But there is the fact they're not going to be as rigid as a protein, because they cannot be. That's an interesting question to go forward. Peter, you want to comment?
Well, I'll first answer as a mathematician. When I was a child, I was a mathematician, and then as a scientist. Whenever you have an interaction that depends on sequence number and is consistent, that always has a tendency to lead to helices if it gets strong enough. And that was the original argumentation with bin Zhang. We were completely agnostic as to causes. Over the years, the connection of that helix formation with motorization has sort of appeared as a sort of folkloric interpretation. And it's somewhat strange, actually, but it's probably true. But you see, when you saw, one of the problems is this extrusion picture. You've seen many cartoons with extrusion complexes falling down. And it's very hard to see in that case why that forms anything really like a helix except for excluded volume. But the densities are not that high. So it's very hard to really see why that forms helices. I think that what happens is that the extrusion complex is not an extrusion complex, but it works by grabbing things and pulling things in, grabbing things and pulling things in, just like Decker sees. And when you do that, that gives rise to an interaction that depends on sequence, not on just complete proximity. So that sort of grappling motor explanation naturally gives it, and my current postdoc, Zhiyu Cao, has actually shown in models based on that you generate helices as you make the motor stronger. You go from an interphase-like state to a mitotic state, and there's various different kinds of things depending on exactly the characteristics of the motors, whether they have very large ranges or small ranges, things like that.
Just to add one more comment to that, I think it's kind of cool. If you remember the plots, I'm not going to go there. The plots we were showing, when you start to get more mitotic, you don't have just the normal single power law, you have two power laws. That tells you that you need some long-range motorization or something like a long-range motorization on the model. In the model, when we just fit it, this just comes by design, right? Because we're just learning from the data when he's inverting it. Most of these motors, condensate 1, condensate 2, they tend to be local motors. So the question is, what creates this other term? Can be the grabbing, can be, okay? It can be that there's a motor that's actually not just acting on the chains that are locally together, but it is the same motor, condensate 1, condensate 2, but when you start to collapse that enough, now it can grab a chain, right? The same motor binds here, instead of picking up on the same chain, picking up on the side chain. I don't know. That's a strong possibility because now the chain is coming closer, right? So you start with some level of compactation. But there's a very interesting question. At least from the data, you need at one point to start to have some higher order term to go there. So but these are very good questions, and I think goes a little bit with the grabbing or density or something else to do this. But the data tells you in an agnostic fashion that you need something like that, okay?
Any other question? So let's thank them, José, for the wonderful talk.